Good afternoon, Grizzlies. I'm Miss Bergschneider, and what we're going to be doing today is we're going to be running one of the experimental conditions for our photosynthesis lab, where we are designing a Biosphere 3. So seeing if we can get a Biosphere 3 to run successfully on Mars. And our guiding question for this activity, so how will you maximize photosynthesis in Biosphere 3 to ensure that the astronauts do not run out of food or oxygen? So we ran our control lab with spinach, and that is going to be the data that we are comparing all of our experimental data to. And in class, you should have learned about some of the factors that affect photosynthesis. So we have temperature, oxygen concentration, light quality, and so on. What we are going to be doing in our test today is we're going to be looking at factor number 10. So we're kind of doing two things here. We're looking at a different producer, and it is a different producer that has a slightly different complement of chlorophyll molecules. So let me show you what we're going to be testing. So we ran our control lab with spinach, and what we're going to be using today is we're going to be doing this experimental lab with this plant right here. So this is called red leaf lettuce, and you can see that it is green, so it has chlorophyll in it, but it also is kind of this purplish color. So it has a different type of pigment in its leaves. So pigments. There are three types of pigments that you will find in plant leaves. So this first one is called carotenoids, and these are long-chain, water-repelling pigments, and they're found in the plastids of plant cells, which I know that probably means nothing to you. So a common carotenoid is B-carotene, and this is found in a lot of flowers. So this sunflower right here, its petals are yellow due to the B-carotenes. What I learned in college and what has stuck with me for years is that your orange carrots you eat, those are filled with carotenoids. All right, so that's the first type of pigment. Second type is chlorophyll, which I know you've learned about in class. Chlorophyll is that green pigment that absorbs light, and it is contained in your chloroplast. And you may or may not already know this, but the wavelengths of light that are absorbed by chlorophyll, so if we look at this chart where this peak is, it absorbs blue light, and it absorbs the red, orange wavelengths. It does not absorb green light, and that's because it reflects it, and that's why it looks green to us. So our producer we are testing today has chlorophyll in it, but it also has this other pigment called anthocyanins. So anthocyanins, these are these water-soluble pigments. They form the pink, red colors that you see in flower petals. And when all of our leaves turn beautiful orange-red in the fall, that is the anthocyanins you are seeing. Now, the interesting thing about this is if we look down here at this absorption spectrum, anthocyanins, they absorb light, so if I use my cursor, in the blue-green wavelengths. So this pigment is absorbing some light in the wavelength that chlorophyll is not absorbing. So it's going to be really interesting to test this plant and see, is it a better producer? Does it produce more oxygen than spinach? Do we maybe want to use this guy on Mars rather than spinach if I'm trying to maximize my productivity and it can absorb more light wavelengths? So that's what we're going to be looking at today. So just like before, I have my biochamber. I already weighed out my 40 grams of red leaf lettuce. So I'm just going to place it in the chamber. All right, we've got it in here. We are going to Stick the lid on, make sure that is nice and secure. All right, so I'm getting closer. What I'm going to do next I want to make sure I have everything ready to go. So I already have my probe on and it is calibrated. If I look at my settings, I have my CO2 probe in. I'm collecting time. It's time-based. I'm sampling every minute, 1 sample per minute, and I'm going to go for a total of 30 minutes. If I look down here, I have my oxygen gas and it is set in parts per 1,000. So that's a unit we're measuring. And I also have a temperature probe on here just because we would like to know the temperature because it affects the rate of photosynthesis. Okay, so my last step is I'm going to seal everything up. And ideally, I would want my temperature probe inside the chamber. However, I didn't have the right size of stopper. I'm going to plug in my light source. Maybe. And hit collect. All right, so we have 1 measurement in, and we are simply going to watch this and let it run for 30 minutes. And then what I will be doing is I'm going to share the completed data set with you. All right, so we're back and we finished our 30 minutes of data collection. So here is the resulting graph. You're going to be working on graphing this in Google Sheets, and our data is over here to the right. As you can see, if we look at time 0, we started at 201 parts per 1,000 for oxygen concentration in our biochamber, and then over here at 30 minutes, if we scroll down, we ended at 219 parts per 1,000. Now, I just want to point out this weird little blip right here in the middle of our graph. It goes up and then it goes right back down. So when you get a strange little blip like that, and it looks like it happened probably about 12 minutes in. So when you are graphing your data and you get to time 12 minutes, it goes from 214 to 218 back down to 216, and it doesn't reach 218 until about 9 minutes later. What that tells me is this is just a little sampling error. So all of the equipment we use, they're going to have little blips, little errors that happen in time. This would be an outlier, and I would actually cut that data point out of my data. So it would go like this in a smooth line and get rid of that. All right. Hope you're having a great day. See you later, Grizzlies.